This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 505, recorded on August 3rd, 2018. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
Kind of hot and muggy here in the city, right?
Oh, it's awful.
No. It's lovely.
No, it's
Love
awful.
it.
It's mostly cloudy. In fact, right now at the window, there are no patches of blue.
Every day we get a little storm passing.
It's
Remember
very
the other day
tropical.
were you in here when all of a sudden a cloud came through, it poured for 10 minutes, and then we looked down and it was sky blue.
Exactly
Amazing.
Right, exactly right.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here in the tropical rainforests, deep in the tropical rainforests
That's right.
of Western Massachusetts.
Still.
Exactly.
Yes.
You
It's cloudy, 86 Fahrenheit, 30 Celsius. Dew point is 71,
Oof.
which tells you that it's sticky. And
Yeah.
and like every day for the past, I don't know how many weeks, it's a chance of heavy rain showers in the afternoon. And
Exactly.
here we are.
Are you feeling like you're a neglected tropical Massachusetts?
No, not really neglected, just but very tropical, definitely.
Exactly.
So joining us from Bend, Oregon, Rich Condit.
Hi, everybody. So we got here in the high desert of Central Oregon, 68 degrees Fahrenheit, 20 Celsius,
Perfect.
35% humidity, clear blue sky. It's gorgeous. It was near 40 Fahrenheit when I got up this morning. Couldn't be better. So eat your hearts out.
Yep.
From southeastern Michigan, Kathy Spindler.
Hi, everybody. I'm trying to figure out what our weather is. It's partly cloudy and 81 degrees Fahrenheit, which is 27 Celsius.
And from Madison, New Jersey, Brianne Barker.
Hi, everybody. It's about 29 Celsius here, so 83 Fahrenheit, and our skies are getting pretty dark. I think we're going to be getting that storm in a few minutes, so I'll let you know so you have a little bit of a warning before it gets to you in New York.
Then we can see.
Actually, we
how long it
predicted
takes
your
to-
storm last time from here.
You
did.
I think she had it first.
So it,
Yeah,
sounds
I-
like,
we can see it first here.
it sounds like New York City has do the right thing weather, right?
Yeah, exactly.
So, folks, it's August,
It is.
and I always find it sad. You know, June and July went poof
Ba-boom.
like that.
It did.
August, that's pretty much it for summer.
Oh, no.
Yeah.
Summer doesn't end until September
Yeah, I know, but
21st.
psychologically, for me, it's
Yeah.
it. History,
I hear you.
N-
Right.
zilch, nada, nothing.
Yeah, classes start August 27th, so I think summer
Oof.
ends in August.
In fact,
Yeah.
I think next week the medical students start to move back in.
Yeah, they do.
Well, before we start talking about science, we would like to ask you to support the Viruses and Cells Gordon Research Conference. The ability to raise money to support this meeting has declined dramatically. Next year's meeting, the organizers, Julie Pfeiffer and Britt Glounsinger, need about $70,000 to support registration and travel costs for speakers, discussion leaders, students, and postdocs. You can go to the secure donation site. Just go to microbe.tv/TWiV, and the latest episode of TWiV, you'll see there a link, and you can give them some money. They would really appreciate it.
Yep.
Finally, President has selected a science advisor, and I do not know the individual. His name, he's a meteorologist, Kelvin, which is appropriate, right?
Perfect.
Kelvin Drogenmeier. Sometimes the names are amazing. His nomination has been met positively by members of the scientific community, according to The Scientist. So that's good, right?
That's
Yeah.
good.
Sounds
I'm-
right.
It is.
I agree with the quote in this article from Marshall Spencer at University of Georgia. I'm pleasantly surprised.
Yes.
He says.
Yeah.
I am. I'm surprised that this administration actually appointed a science advisor, and that it's somebody who is not a completely bizarre choice for the position. I don't know if this is somebody who's going to be listened to at all, but it's a good development.
It's a good step.
The hope is it's a meteorologist who's not a climate denier.
Oh, I think this is a legit. I think this is somebody who actually is in the science.
He's according to this article, a well-educated individual. Okay, I mean, he's a genuine scientist.
What was Trump thinking? This is totally counter to everything else he's ever done at that level.
That's probably the wrong question, but we should move along.
All right.
Clarissa writes, Dear TWiV team, thanks for the discussion on the challenges of polio eradication. It was great. Regarding the Brazilian vaccination program, you're right, it's great. An outstanding public health action that we have had for free since the 1970s, with increasing number of vaccines added over the years. But Brazilian people take it for granted. And not only for kids, but for teenagers and adults, such as HPV, HBV, flu, etc., all free, and we still get low coverage. Too bad. Actually, measles is back 2 years after Brazil got the eradication certificate by WHO. This is so absurd.
I would agree.
Clarissa, of course, is at the Carlos Chagas Institute at the Universidad Federal de Rio de Janeiro.
Very famous place.
A very famous place.
When he says they take it for granted, that doesn't seem to resonate with what has actually happened. Then, if you took it for granted, it means everybody just goes in and gets their vaccine. And
that's
what I would say.
The linguistic
for
term.
That it's available, and maybe I'll get around to it, but maybe I won't.
So they're blasé.
Yes.
Mm-hmm.
Sure.
Yes, they are blasé.
Dickson.
Anthony writes, according to the PR above, many bats were found in the well. The family was drinking water from the well.
Stop. Let's stop there and analyze this. This link. I know there's more, but this is a press release from the government of India.
Right.
May 22nd.
Right.
Which Anthony found. And this concerns the well in the area where it may have begun. The plumber went in to fix it or something. And apparently the family was drinking water, the team found many bats housed in the well from where the family was drawing water, which I guess you would do from a well, right?
Right.
Some bats have been caught and sent for lab examination, and we don't know. So
that's from May.
It's from May. I'm sure they must know about
This, by the way, for those just tuning in since last week, is all relevant to the Nipah virus outbreak that we discussed last week. And where did it come from?
Yeah, there was.
So this is presumably after the guy went into the well to clean out the bats, and then the Nipah outbreak occurred, and then the government came and investigated and apparently found many bats in the well, whereas another report that we got was that they only found one bat, which was an insectivorous bat that was not carrying the virus.
Okay. Go ahead, Dickson. Proceed.
Was Dr. Peter Reed, the Australian vet with Hendra experience, who emailed, correct. TWiV. I was not able to find the original TWiV letter by a search. Yes, indeed, he would most certainly be an interesting guest. By the way, in that TWiV letter of a past episode, Dr. Reed asks about American fruit bats and disease. These are New World bats that eat fruit, but alas for the curse of common names, these are not the kinds of fruit bats that were referred to. The American fruit-eating bats are Microchiroptera, not Megachiroptera. If memory still works for me, the Artabeus are related fairly closely to the vampire bat, which that's not a fruit-eating bat. That's a blood.
So Peter Reed.
It actually doesn't suck blood.
Right. So these New World fruit bats are not called fruit bats.
Right.
Apparently, that's the common name problem. Even though
vampire bats don't suck blood. They do not suck blood.
What do they do?
Did you ever see them feed?
I never saw any bat feed.
No, they've actually photographed these with infrared cameras. The cattle sleep standing up, and the bats creep up on the backs of their necks, and with their teeth, they slash little cuts in their hide, and then they lap the blood up with their tongue as it drips out. They don't suck it.
Okay.
So
they do drink it.
Then they say, I want to suck your blood. That's a human vampire, not a bat vampire.
Bela Lugosi.
Right.
Mm-hmm. And in fact, it was Peter Reed. That was the vet. I went back and dug out TWiV 41, his first letter. Love your show and keep up the good work. TWiV 41.
That's
by way of quick introduction.
Going back.
I was the vet involved with the outbreak and deaths of 14 racehorses back in 1994 here in Brisbane, Australia.
Right.
And he goes on, and then he wrote back on TWiV 93, which is in 2010. And the key here was, let me know when you're planning to do the show, and I'm only too happy to talk with you on Skype. Keep up the good work.
Outstanding.
So
we should do that.
We should do it.
We should.
Definitely.
Sure.
He's still around because he has a paper this May, actually, which we'll get to. It's actually cited in the next letter. Alan, can you read Colmes, please?
Yes. So Colm writes, for once, I'm mostly caught up on podcasts instead of a week or two behind, and thought you might be interested in this article from The Atlantic recently about the anti-vax movement making it to horse farms in Australia, and he provides the link.
Last week,
buddy at UT Medical Branch.
Last week we talked about the Brits who don't want their dogs to get autism, so they're not vaccinating their pets, right?
I think I mentioned that I had read an article about horses and Hendra, and this is the article that I'd read.
He read your mind.
He did.
It's The Atlantic. Anti-vaxxers are targeting a vaccine for a virus deadlier than Ebola. And it's just, I don't want to put strange substances into my horse, and I don't believe in injecting chemicals into horses. Okay. And then some people are afraid.
So if the horse gets a bacterial infection, you won't give it antibiotics.
No, that's a strange. I don't do it.
So
sorry.
Some of them are concerned about the performance of the horse. These are race horses, after all. Okay. So there's an article linked to within that article. It's a Australian Veterinary Journal article in April of 2018, investigation of the effect of Equivac Hendra virus vaccination on thoroughbred racing performance. Thoroughbred is with a capital T. I didn't know that. It's a regular.
It's a proper name.
Anyway, that one of the authors on this is Peter Reed. And there's no effect of being vaccinated or not on any of the things they measured. And this is kind of interesting. They have 4,200 race starts by 755 horses. With or without vaccination, they compared the time form rating, the margin to winner, the prize money won, wins, and places.
Come on.
Wonder if they spent a bunch of time at the track.
Do you think?
Sounds like it.
Yeah.
No effect of the vaccine. That's great.
So this is complicated. This article is actually very interesting because this is it's complicated because a lot of veterinarians won't treat your horse in particular if it's showing symptoms consistent with a Hendra infection if it's not been vaccinated, and I don't blame them.
Yeah, it could kill you. Yeah.
Yeah. So it's complicated, and that article is very good.
The weirdness just multiplies.
Yeah.
I have to ask Anne Simon if she lived in Australia if she would vaccinate her two horses. Anne Simon was the local host for ASV 28.
Yeah,
I can't believe she wouldn't. Actually, the other issue is apparently the vaccine is pretty expensive, and it's got to be constantly boosted, if I understood this correctly. So it's a complicated issue, but still the anti-vax sentiment doesn't make any sense at all.
No,
not at all. Hendra seems to be pretty bad for the horses.
Not good.
I think that you'd want to protect against that.
And it's one of these things like the HPV vaccine where it's just a protein. There's no virus or anything else.
Right.
It's just a protein.
Kathy, can you take Ted's, please? She left.
No, I'm here. Sorry, there's Ted. Sorry. Dear TWiV hosts.
Hi, Ted.
Do we know Ted? Oh, yes, we do, I guess. All right. Dear TWiV hosts, thought this recent Lancet article, paywall, and commentary in the New England Journal of Medicine Journal Watch might be worthy of mention as a snippet in terms of the apparent long persistence and transmission of the Ebola virus by a female in one Liberian family. Congratulations on reaching your milestone 500th episode. I'm eagerly anticipating the 1000th. Keep up the excellent advocacy for science, communication, and truth. We, your listeners, will accept no substitutes. Ted. And
Great.
Ted is from Fort Lauderdale, Florida.
So, yeah, Ted has written many times. He said, DMD. We have a few listening. And it's good to hear that clacking noise in the background. And the paper that Ted suggested was published in Lancet Infectious Diseases, Persistence of Ebolavirus After the End of Widespread Transmission in Liberia, an Outbreak Report. And this comes with 4 joint first authors.
Followed by a cast of thousands.
And then 2 contributed equally last authors. And the 4 joint first authors are Emily Dokubo, Annika Wendland, Suzanne Mate, and Jason Ladner. And the 2 final authors are Gustavo Palacios, who has been on TWiV. I don't know if anyone remembers. No, nobody remembers.
The name sounds familiar.
Yeah, he was on with Ian Lipkin a number of years ago. He used to be here in the Lipkin lab. And Mosoka Fala. And there's a connection here.
Right. And I was just reading the response from her when you asked me to read Ted's email. So that's why I was a little off and distracted. So Kaine Dokubo is the first author. She goes by her middle name of Kaine. And she was a summer undergraduate in my lab at the University of Georgia in 2000. She's a student at Xavier University in Louisiana at the time. And then she went on to medical school. She got her MD from Emory University, and then she got a master's in public health from Johns Hopkins.
Wow.
And according to her official CDC bio, then she spent time in San Francisco, and she has board certification in internal medicine and I'm trying to find it now, preventive medicine and public health. And so she was the, as I said, the first author on this, and she's the corresponding author. And so I was lucky because there's only one email address of all the long author list, and it happened to be hers, and it got right to her. And so I was able to confirm that, yes, indeed, she's the same Kaine Dokubo who had been in my lab, because I never knew her as Emily. And she's always Kaine.
Interesting that she used her full name then.
Yeah, I think, maybe the Lancet did it that way. That's how it's listed on her CDC bio. But, there's lots of people in the world that go by their middle name. And
I would never go by my middle name.
What is her country of origin?
Well,
Nigeria?
If I remember correctly, she was born in London and then lived in Nigeria for a good long time. Mm-hmm. And then her parents... I think at the time when she was in my lab, they were living in Georgia and she was in Atlanta, but she was going to school in Louisiana. But she was commuting from Atlanta that summer. And now she's the country director for CDC Cameroon. And so evidently she and her family live in Cameroon and in Georgia.
Cool.
So I'm not sure what the country director means. Let's see. Here. CDC has field offices in over 50 countries, working primarily under the U.S. President's Emergency Plan for AIDS Research Relief, which is PEPFAR, and GHSA. I'm currently assigned overseas in Cameroon, and I travel back to the U.S. for work and meetings. So, her parents were in graduate school in Washington, and she returned to the United States for high school. And English is the official language, so that's how she got her schooling. And she understands local Nigerian language, Igbo, Pidgin Creole, and a little French. So mostly English, I guess.
So this comes from the CDC in Atlanta, but also WHO in Monrovia, the U.S. Army Medical Research Institute of Infectious Diseases in Frederick, which is where Gustavo and a few others are from. NIH in Bethesda, the National Public Health Institute of Liberia, International Rescue Committee, Monrovia, the Ministry of Health in Monrovia, and the Pathogen and Microbiome Institute in Northern Arizona University, Flagstaff, Arizona. So everyone may remember the 2014-15 Ebola virus outbreak in West Africa. We talked about it many times.
We had a lot of TWiVs on it.
It was a long arc.
It was.
Began in March 2014 and ended kind of mid to late 2015. And this paper deals with a cluster of cases that happened after the epidemic was declared over. And it has to do with persistence, possible persistence of virus in people who have recovered.
Right.
Now, we talked about a couple of these cases before, and in fact, one was a paper on which Gustavo Palacios was also an author. It concerned transmission by sexual intercourse from a survivor, a male survivor who had recovered and months later apparently transmitted it, and a few others. So this cluster happened in 2015, which was just after, in May, Liberia was declared free of transmission, and then this cluster of cases happened in June.
So it raised the question of plaque assay versus antibody. And plaque assay would have been much better to have here.
Very hard to do.
Right? Well, or demonstration of the virus
because
itself.
You need a BSL-4 if you're going to do a plaque assay. Yes, Kathy.
You said that before this outbreak in June, but actually this outbreak was in November.
Yes, I know.
Yes, and that's actually when Kine was deployed to Liberia.
Mm.
Right. Yeah. So there. So you just ran through a series of events here. The 2014 to 2015 outbreak, and then there was a cluster in June 2015.
Right.
That was transmitted from an apparent persistent infection, and then the cluster we're going to talk about in this paper is November of 2015.
Right. And remember, in May, they said Liberia's disease-free. So
right.
They had a June outbreak, and then they had a November. And after November, then they said it was disease-free, and it, in fact, has been since then.
Yes.
But this cluster, so this is basically a case investigation of this outbreak cluster to see what happened. And it starts with an index case. It was a 15-year-old boy, and this is individual A. You're going to have to remember the letters. They're all given letters.
It starts to get a little confusing when you're going.
Yeah, there are a lot of letters.
So, is this paper open access?
Don't know.
No,
it is not.
No, paywall. Ted said paywall,
it is,
right?
And I confirmed it's a paywall.
So this person, this 15-year-old boy got sick. He was diagnosed with Ebola on November 19th, put in the Ebola treatment unit, and then he had a father, a mother, 3 younger brothers. Since they all lived together, they were put in the ETU as well. The father and the 8-year-old brother were positive for Ebola RNA. The mother, the 2-month-old brother, and the 5-year-old brother were negative. So the key letters here, the boy is A, patient A. The mother is patient D. Those are really the important ones. Unfortunately, the little boy died at the end of November.
The 15-year-old.
The 15-year-old boy who started all this. Father was seropositive IgM and IgG for Ebola-specific antibodies, and they say this suggests he was infected before his sons. The mother had IgG and a low amount of IgM in her blood.
And she has an astronomical IgG titer. I've never seen anything like this.
Very high titer. Very high titer.
Yeah. Very high.
So they said, where did this infection come from? They tried to rule out all possible sources. No travel to Ebola-affected areas. No parts of transmission chains, no contact with someone who was sick, no funeral attendance, no sexual contact with known survivors. So they basically ruled everything out, and they said, well, maybe it's persistent infection. So they investigate the mother. In July of 2014, she took care of her sick brother. This is person G. We're moving along the alphabet here.
Yes.
This person, her sick brother, worked as a nurse's aide at a local clinic, and this person, individual G, and his supervising officer took care of a patient who had symptoms that looked like Ebola and eventually died.
So the patient was individual X, and the supervisor was individual H. In case you want to keep track of those.
So G and H. So G is the brother that the mother had cared for. They both had Ebola. And G and H both died. So this is Ebola here in this little cluster of cases.
But remember, she took care of them in July 2014.
Right.
And the rest of these cases are showing up November 2015.
Right. So it's more than a year.
Now,
and she, after taking care of them, she became ill with symptoms that were consistent with Ebolavirus disease, but she didn't go to the hospital or get tested.
Right.
Right. So she was never formally diagnosed.
Not diagnosed.
Right.
In the meanwhile, she was also pregnant.
Right.
And she miscarried in August of 2014. She recovered. And that's the end of that. And a year later, roughly, September 2015, she gives birth to a baby boy. Okay. And then a little bit later, October 24th, she gets sick. She has 2-week history of fatigue, etc. Looks like Ebola, but they didn't actually diagnose Ebola in her. They didn't take any blood from her.
Right. They treated her for anemia and malaria.
And she got better, and she went home. So they're thinking maybe she had Ebola twice, right? The first time, and when she miscarried, and then a year later, roughly, after she gave birth to her child.
And the idea would be, the hypothesis is that she was carrying the virus all during that time.
Yeah.
And so recrudescence, the virus at least the symptomatic infection shows up again after a year. And it's at that point when she would have given the virus to her husband, who gave it to the son. By the way, if any out there who can get this paper during this whole discussion, and I'm sure you guys are doing the same thing, I'm looking at figure 1.
Yes.
Okay, which maps all this out once you get into it, maps it all out in a very nicely understandable fashion.
Yes.
And it reads a little like a detective story.
Oh, it's great.
Yeah, it does.
Yeah.
Now, they didn't take a blood sample from the mother in 2014.
Right.
So it's hard to, they have subsequent blood samples, obviously, because that's why we know
but she didn't seek medical attention in 2014.
Yeah, so we don't have a blood sample. So they're using the other members of this cluster, H, I, and J, who did give blood, kind of as a proxy to say what happened to her, because these cases all occurred at the same time. So they have genomes from some of these individuals, but not from her. But again, she's part of this cluster. So they do some genome sequencing, and they can see
Ebolavirus genome sequencing.
Ebolavirus, not human genome sequencing.
Right.
No. They could get that from her, obviously, because she's still around. Ebolavirus sequencing. And the genomes group with a sublineage that was circulating in Liberia around August 2014. So now this is 2015, right? This cluster of cases. But the virus looks like it had been circulating a year ago. And the idea is that it's been persistently infecting this individual and not undergoing a lot of genetic variation.
Right.
And so you have this sublineage virus, and they also have changes that they identify in particular isolates that are unique to them and not present in the other members of the lineage that had previously circulated. So this gives some support from the idea that, this was a virus that she got in 2014, persistently infected her, and then recrudesced in her and infected these individuals, and then that's why this lineage looks like it was around in 2014 and not 2015. So,
and the idea is, since pregnancy is immunosuppressive, that may have been what triggered the virus to recrudesce.
Right.
So the question, of course, is where did it persist in her, and how did she transmit it to her family, right, to the boy who started this case and others? And we don't know. But
the first question is a lot more mysterious than the second. I mean, these people lived in a household together, so it's close contact.
Yeah, they say it was close contact. There is, and they say there's no recorded evidence of sexual transmission from female survivors, but most likely transmission occurred during close physical contact.
And aside from persistence in semen, the only other place that we know of persistence is in the eye. Is that correct?
Right.
Correct.
Well, they say also breast milk and cerebrospinal fluid.
Okay.
Right.
So I would say eye is a possibility, or breast milk in her case, I guess. Not I don't think she's shedding CSF, so that's not it's not an issue that we have to
usually eye, placenta, testes, and CSF are kind of thought of as the big immune privilege sites. Mm-hmm. So those would be places to think of.
So given the number of people exposed and the number of people who died from the original outbreak in 2014, is this a rare occurrence, or are there
rare, yes.
Are there other expected occurrences?
They say they make that point. They do say it's pretty rare. We've got less than a handful of similar cases, right? So in the big scheme of things, yeah, it's rare.
But
thank goodness.
The accompanying commentary points out that 17,000 survivors now exceeds the total number of survivors from all the previous Ebola outbreaks combined.
Right.
So we're starting to have more numbers along those lines. And that the fact that it might be difficult to always diagnose Ebola, even at its peak or after its peak, that the number of survivors could be underestimated. And as they say, this is supported further by the observation that reemergence can be regarded as a rare event. So the fact that we know of this, at least at this one reemergence, that there could be a lot more.
So wouldn't you, as the EIS officer from CDC at least, want to get those 17,000 people diagnosed now to see whether or not they're harboring latent infection?
Well, where would you look?
Cerebrospinal fluid.
No, that's
you're going to got a big list here, boy.
I know, but
talking about West Africa.
That's hard. That's hard.
We're going to go to West Africa and do 17,000 spinal taps.
No, not spinal taps. You could do, semen for sure. You could do blood. You could do
from half the population.
Half. That's right.
Yeah, I'm not even sure. You really don't know where to look.
What does it tell you?
It tells you that those people might still be infectious.
As they say, you have to be vigilant about looking for symptoms, and they say this part of Africa has now learned how to respond to outbreaks. So that's what you do: be vigilant, and if you see something that looks like Ebola, you go right in and you take the family, as they did in this case, put it in an ETU.
So could any of the other outbreaks of Ebola have been caused by this rare kind of a reemergence?
Any of the other?
Yeah, we've had a lot.
The evidence suggests that every one is a brand new spillover because you sequence the virus and you can see it's not.
That's true.
So even the current one in the DRC is different from this one.
Yeah, I'll take that.
Right?
Yep. Mm-hmm.
So I suspect that it's going to be rare that you're going to have this kind of recrudescence, but you got to look for it.
So given the genomic analysis of the Ebola virus in this particular case, are you now hypothesizing that the replication rate of the virus is very low in those individuals, and therefore the accumulation of mutants would be slow?
The outbreak involves a handful of people, so you're not going to get the diversification that you would in a population for over a year.
I mean, it's not a raging infection in these people. It's been kept under control by, like Brianne said, in these privileged sites. So the replication rate would be very, very low, and therefore you'd expect the strain to stay the same throughout the time.
At least with some DNA viruses, when there's persistence or latency, it might not even be that the virus is replicating at all. Mm-hmm. So.
Yeah, I mean, with RNA viruses, it's hard because this is not a typical presentation for most, right? So we're not quite sure when the virus is in the eye for months. What is it doing? Is it just sitting there?
Exactly.
So I guess it could be, but that runs contrary to my training. An RNA virus is just sitting there.
It can't just sit there.
So to Dickson's point, the effective replication rate might be slow, but maybe it doesn't replicate and then it replicates in a burst or something.
It has to be still within inside of a cell, though, correct? It can't be in the vitreous humor or something like that.
Why not? Sure.
Yeah.
Won't RNases degrade it then?
Well, that's why I said it's kind of contrary, but it could happen. Why not?
If it's encapsulated, I mean, if it's not just RNA, if it's the virus.
It could be covered with amyloid.
Or mucus.
Right.
Right. Yeah, and another aspect of this is that you've got all this apparent transmission. You've got several incidents of apparent transmission of this from the mother, and not everybody got sick, and only one died. What I find especially interesting is individual E, who's the 2-month-old baby.
Mm-hmm.
who's alive, no symptoms, is Ebolavirus IgG positive, probably because mom is absolutely chock full of Ebolavirus
Thank goodness
IgG.
He's not IgM, right?
And giving it to it in breast, and he doesn't have IgM.
Right. So that could be from the placenta too.
It could be from the placenta, right? Or it could be that the breast milk is protective.
So as Rich said, figure 1 is great. It shows individual G, which I'm sure everyone remembers, was the brother who was sick, and the mother who's D. So G transmits to the mother who's D, who then transmits to her husband. A year later.
Transmits how to the mother?
Well, she was caring for his sick brother. So he was presumably shedding, and she's close to him, caring.
Secretion.
Bodily fluids. Close contact with bodily fluids, which happens under medical care.
Yeah.
All right.
I don't think you would dispute that.
No.
And then, then transmitted into the family, including the 15-year-old who started this all and died in November. The other child, the 8-year-old, survived.
The scenario that they postulate at the end is that that it was transmitted from the mother to her husband, and then that was somehow transmitted to the children.
Yeah, that's figure 1. It shows that too. Yeah.
And the 2 children, the 2 older boys, that is, both received ZMapp antibodies. So that NIH already had approval in place for that. So they got it a few days after they were admitted to the Ebola treatment unit.
So I think that this is, it's interesting and important just phenomenologically, but also, as we've already said, it seems to me that historically, most of the Ebola outbreaks have been, or all of them, have been spillovers and relatively rare. And now we have this big pool from the 14-15 epidemic of survivors. And there's although there's, we don't know the percentage, presumably pretty rare, there is the possibility of persistence. This kind of thing could become a major pool for outbreaks in the future.
If you were the health officer for that area, would you identify those people, the 17,000 survivors, and say you may not donate blood? Would you say that?
Yeah, based on this,
that's a consideration.
Probably worth thinking about. Yeah.
I mean, the survivors. Yeah.
And there was blood transfusion in part of this story. I can't remember exactly where, and they went back and they checked both of those donors, and they were negative.
Right. So I think it was that the mother got a transfusion.
The mother got a transfusion when she was
her second illness. Yeah.
Right.
So that is the real question: is how long does the persistence go? Right. So we don't know. We'll see. I mean, that's
how common is it?
Yeah. So the 17,000 is going to be an interesting cohort to study. But as they say, here's the sentence: the rapid containment of flare-up associated Ebola virus disease cases indicates the increased public health and outbreak response capacity in West Africa. So they'll be on it.
So there's something else to think about here too, and that is Kathy's remark about pregnancy being an immunosuppressive event. And maybe that triggered the event that resulted in this woman's.
That's the idea.
There are lots of other immunosuppressive events possible, malaria, for instance, and lots of other infectious diseases as well.
HIV.
Yes, exactly right.
Measles.
So now you've got 17,000 potential epidemics on your hand that could eventually prove to be the sentinel case for a new epidemic.
We'll see. We will keep track of them here on
this
the
is fascinating.
It's different. It's a different situation also, because of all these survivors.
Depressing.
Right. And I asked Kaine what was kind of the most interesting or exciting part of this, and what kind of lessons did they learn? And obviously, as she said, the most interesting was that there had not been evidence of a female survivor possibly being persistently infected and transmitting it. And the obvious lesson is continuing that Ebola virus persistence means that you have to be continually doing disease surveillance so that you can catch these things rapidly. The thing that the commentary pointed out that I thought was interesting was the fact that there will be stigma for these 17,000 survivors. And so how to handle that is quite important. And so they had, guidelines at the time. And then in this commentary, they allude to the fact that WHO is working on them. And Kaine said that new guidelines have not yet been released.
So another comment to make here, too, is that Lassa fever, which is not a related virus, obviously, those survivors donate serum as a backup for stemming outbreaks. You can actually use the IgG fraction to prevent death from the infection. Where in this case, you'd say, well, it's just like Lassa. Let's take some blood from these people and stockpile it in case there's a new outbreak. We can just use their IgG. But you may be actually transmitting the virus.
Well, you'd have to check if you, obviously, right?
Obviously.
You could check very easily the serum. So they mentioned few occurrences of recrudescence have been reported, and one of them is uveitis. Wasn't that the fellow we met after TWiV?
Yeah.
It's Ian Crozier.
Ian Crozier, who was a doc working in West Africa, right? And then there was a Columbia ER doc also who came back, and he went bowling in Brooklyn, and the next day he developed a fever, and they hospitalized him, and he had recrudescence as well.
I believe there is a nurse from Scotland.
Yes.
Who has had recrudescence. She's had it come back a couple of times.
That's right. And we've talked to the doc who took care of her in Scotland. Right. Emma Thompson was her name and still is. And we talked to her after the first, she talked about how they cared for her initially.
Wow.
Ebola Experiences. That was the name of it. Okay.
One thing that I thought was interesting was that this, happened in November of 2015. They collected specimens 6 months later for additional serologic testing. And so I asked Kaine, because the Lancet doesn't give you submission and publication dates as to, did it take a long time to write it, or did it take a long time to get it published? And evidently it's the former, because they submitted it in June and it was accepted in July of this year. So about a month after submission.
So if I was rewriting this story in Alan-esque language, I would say Ebola bowls a gutter ball.
By the way, there is a new Ebola outbreak in the DRC.
Yes.
Brand new, just started, just as the last one ended, which we talked about a couple of weeks ago. There is a new one. It's killed 20 so far, 9 days after health workers declared the virus contained. Now, this is in the North Kivu province, a different area.
And we still don't know the source?
That's probably another spillover, I would guess.
Yeah, from where?
The forest.
The forest.
No, that's not fair.
What do you want to know exactly?
Yeah, you want to know what about that forest?
Well, most of the spillovers, they don't know the...
Bats or something like that.
I mean, they say there's some contact with bushmeat, but sometimes there's no known contact, it's hard. You think it's hard to get CSF?
Sometimes it's
17,000 people. This is very hard.
No, I got it.
And this is an area of considerable unrest, which makes management of all of this very difficult. Exactly. This is close enough to the last. Seems to me that it's close enough to the last cases. Well, it's geographically remote, right? I'm wondering if it is necessarily a new. Yeah. It doesn't have to be a new spillover. It can be more of the same.
Could be.
I think they said it was pretty geographically remote, sort of like the opposite corner of the country.
I'd call that whole country
We'll see.
remote.
They'll get some sequences and we'll find out.
Yep.
And you will hear it here. I wonder if they're going to bring in vaccine like they did for the other outbreak.
I believe, yes. I read an article today that says I think they're already using the vaccine.
Well, it says here, maybe your article is more recent. Mine is 2nd of August. This article, the Telegraph, says it's unclear if the vaccine can be used because it's not yet known which of the 4 strains are involved in this. And the one that the vaccine has Zaire.
There's no combination vaccine with all 4.
There is no combination, no. There's just the one. Developed in Canada and spread by Merck. What do you call it? What's the word?
By Merck.
Distributed.
Or disseminated.
Spread as a bed or dissemination. Yeah, that's right. Vectorized.
Because last time someone corrected us, it was developed in Canada by New Link Genetics, I think. All right, go back to the last email. All right, we have a paper which was suggested by Prasad.
Just so you all know, about 15 minutes ago, it started to storm hard. So
we can tell from here that should have been the case, by the way.
Well,
so you should have some more.
Let's see how long it takes.
We can't see your house from here.
No, here, Dickson, here. The rain pattering against the window.
Vincent, I knew what you meant. I'm baiting you with this now.
Dear Vincent and the other TWiV hosts, great job continuing the wonderful TWiV podcast. Love the term someone coined this week on Digisphere to describe Vinny as the podfather. I wanted to bring your attention to two papers that I noticed this week. Once again, suggest pivotal roles played by viruses or virus-derived elements in human existence. One is a paper in Cell reporting that a line 1 retrotransposon regulates early development. The other is a paper in Neuron that is highly suggestive of herpesviruses as the underlying cause of Alzheimer's disease. I was blown away by each of them, although the latter still needs to be confirmed to show that herpesviruses are not merely opportunistic passengers. It's a great story. If true, one could use antivirals to stall them, if not to eliminate them completely. We'll get back to that. On another note, I heard on the last Immune that Vincent is beginning to worry about the future of these podcasts that have become such favorites for myself and a host of others I know, including my students and postdocs. I dearly hope that they will continue and that you will find the resources needed. In the meantime, let me toast your recent successes, TWiV 500, Vincent becoming the podfather of the science podcast genre, and the TWiV subscription exceeding 13,000. With regards Prasad, by the way, while my full name is Vinyaka Prasad, everyone calls me Prasad, not Vinny. My real last name is not in my name. Sorry about this nonconformity. So you call him Prasad, even though his name is Vinyaka R. Prasad. And he's a professor at Albert Einstein College of Medicine, and he was a postdoc with Steve Goff, and he was on this floor. So I remember Prasad.
How was that?
I don't know.
Many moons.
Not many moons. Maybe 15, 20 years ago.
Many moons before I came here.
Before you came to the floor.
Yep.
So I thought we would do the Alzheimer paper, which was also suggested by Bob, who sent it and said, looking forward to some interesting TWiVs. So it's an interesting paper. Not an easy paper to get through.
No.
It's published in Neuron. And
it is open access.
Okay, excellent. Multi-scale analysis.
Let's just quit there and let them struggle with it.
Yes.
Yeah.
So go for it, folks. Check this paper out.
Multi-scale analysis of independent Alzheimer's cohorts finds disruption of molecular, genetic, and clinical networks by human herpes virus. So this comes from multiple places. We have two co-first authors, Ben Reedhead and Jean-Vianney Auré-Mirand. And then we have the lead contact is Joel Dudley, who I briefly saw at ASV. He's from Mount Sinai. Other people are from Arizona State University, the Arizona Alzheimer's Consortium, the University of Arizona, and the Banner Alzheimer's Institute of
and
Phoenix.
You may wonder why that's down
Systems
in
Biology in Seattle, the VA Medical Center in New York, some place called SEMA4, which I assume is a company. And I found this interesting. The Center for NFL Neurological Care.
Oh, that's where the brains come from.
Yeah.
They have brain banks. So this plays an important role in this paper, brain banks.
There are an inordinately large number of people over the age of 65 that live in Arizona.
Yeah. So there are about 30 million people a year having Alzheimer's. Is that right?
Yeah, and they all moved to Arizona.
Right. There were what?
They all moved
a lot of them
Arizona.
are in Florida.
They're all over the world.
The other part of them are in Florida.
Are there any countries without Alzheimer's?
That's a great question.
Didn't you ask me that previously?
I did. Yeah. I did ask you.
So what's the answer?
I have no. I asked the question. Remember,
Are there any countries where nobody lives past age 50 or so?
There are, but Alzheimer's is you can get early onset.
I think that there is Alzheimer's in most countries. One of my colleagues in our biology department does a lot of work with Alzheimer's, and I was very glad for that as I read this paper. And I think there's a big Alzheimer's study among people in Sweden.
Yes.
So it's a chronic neurodegenerative disease.
Right.
60-70% cause of cases of dementia. The most common early symptom is difficulty in remembering recent events, which I have often. And seriously, maybe I have, I'm 65. I'm in the right age.
Everybody there. So there's a spectrum of this. There's normal, occasionally people forget things. Then when you get into the clinical scaling of it, there's mild cognitive impairment, or MCI, which is a point at which you can say, okay, this person is abnormally forgetful. And there's a lot of discussion about whether that is a prodrome for Alzheimer's disease. Alzheimer's disease is the more advanced stage of it and is characterized by severe cognitive limitations and also these distinctive plaques that form in the brain with amyloid beta proteins and tau proteins. And there's a lot of debate about which of these things is causative and which is effect and what exactly is going on.
I believe you can only diagnose them officially upon autopsy, which makes it hard to see what's going on.
Right. You have to section the brain and look for the plaques.
So 60, 65 years and older, roughly, right?
The majority, yes.
There's some early onset early as well.
There is early onset, yes.
And it's named after Alzheimer.
Yeah. So Vincent, what you're suffering from is old-timers.
Actually, no one on this podcast
has
any of the symptoms. I just, I shouldn't joke because it's serious. And obviously, there's so many people. It says here, very good Wikipedia page. In 2015, there were 30 million people worldwide with AD, which is not Alan Dove. It's Alzheimer's disease.
Yes.
Yep.
And so lots of resources being devoted. Many, many companies trying to get a miracle drug. Wouldn't that be amazing?
It would.
If they could have one drug to treat them all.
There are some articles about mice with an Alzheimer's-like plaque formation.
Yeah, you can make them.
There are some drugs that do reverse that, and so they have some recent success.
Mouse experiments in this paper doing that.
There are a number of risk genes that are implied in having a higher risk for developing Alzheimer's, right?
One of the neurologists here was important in that study, by the way. His name is Rick Mayhew.
Oh, I know.
He's the chairman of the neurology department.
But there are a variety of genes that have been implicated, including beta-amyloid, tau proteins, and many others as well. It's pretty complicated. Lots of activity. And so here is a paper where they started to try and figure out, what gene networks were altered in Alzheimer's patients. So looking at transcription of genes and saying, well, what's really different? And they have a resource at Sinai called the Sinai Brain Bank, which is these are brain samples from people who meet neuropathological criteria for AD, but they were cognitively intact when they died. So they call this
So I presume these people died of other causes.
Correct.
Mm-hmm. Yeah.
Okay, and they were okay, but on autopsy,
they find this, right?
Were revealed to have some pathological symptoms of Alzheimer's, but apparently not enough to cause any cognitive problems while they were alive.
It's my understanding that Alzheimer's disease is not a fatal disease. You die from something else. With Alzheimer's, you don't die from Alzheimer's.
Well, you have neuronal degeneration and death.
But then you're bedridden and you can die from pneumonia.
Well, you could, but you could also.
Well, right. And ultimately, everybody dies of a heart attack.
Exactly.
But this is
cessation of heartbeat.
This is a condition that is degenerative and progressive and is going to lead you to a point where you're going to die.
Of course.
But importantly, in the samples that they start with in these people were not cognitively impaired.
No,
that's
right.
So they call it preclinical AD.
Right.
And they can laser capture cells from specific regions of the brain.
It's amazing.
There's some regions that are more where you have more neuronal loss than others. And they can then do RNA sequencing on these cells and ask, what's the transcriptome? And they can compare these preclinical Alzheimer's with healthy controls and see what's changed. That's what they set out to do. So this is really a big RNA-seq project, essentially, in the end, but it's very complicated. And if you can get access to this, you'll be astounded at the figures are Amazing. Yeah. They're so small.
They have the smallest font I've ever seen in a figure.
They are as bizarre as the Alzheimer's plaques.
There's this figure 1 is a microfiche that they printed on the page.
Exactly. Yes.
That details the whole experimental protocol, and so the issue with complexity in this paper, which you can go and look at, is that they're trying to find the signal in all the noise of all the RNA that's in these cells, and so they're doing these combinatorial analyses and trying to work out the network of possibilities that could be involved in development of Alzheimer's disease.
And when they do this first comparison between the preclinical and the controls, they only look at 2 brain regions.
Mm-hmm.
Right.
And those are the entorhinal cortex and the hippocampus.
Yeah. And they find, in fact, networks of genes that differ in preclinical AD versus normal people. And these include transcription factors, et cetera. But they notice that these happen to also include genes that are involved in susceptibility to virus infections, to responses to virus infection. And so because there's a history of people thinking about involvement of viruses with Alzheimer's, they decide to focus on that. And, the way they presented is that they weren't looking for viruses, but they found signatures.
So in figure 1, in C, when I look at it very blown up on my screen, I can see some of the genes that they came up with.
Mm-hmm.
And a couple of them are standouts. The first one in the middle box, ApoE,
mm-hmm.
Yes,
is the most frequently associated gene with late onset Alzheimer's.
Right.
And it looks like that one is actually unchanged between their 2 groups.
Mm-hmm.
And there are a couple in the drivers lost in preclinical AD, the APP and IL-1 beta, that I think are also previously associated with Alzheimer's. So those, I was sort of questioning what was going on with those. But maybe this is because of the specific type of patients they were looking at.
And then in that same, next to it, indeed, they have network drivers that overlap with viral biology, which just is a term that drives me crazy, overlap with viral biology. But anyway, they have genes that have been previously implicated in, as I said, susceptibility to viral bronchiolitis, EBV infection, papilloma infections, etc. So that's what...
And what you don't know from this is how they got from this list of genes to the ones that overlap with viral biology. I mean, did they cherry pick, or did they, just took these boxes, each of which have about 15 genes in them, and just look at those top 15? Or did they look at more and only put the 15 in the box?
Yeah, I was thinking that. I mean, I'm reading the paper and looking at the figures, and I'm taking what they say on faith, because I can't go to the data and check it. It's impossible.
Right.
I don't know how the reviewers did it even, right?
Nope.
Well, so one of the things that I was wondering about, this is, I would call a systems biology paper. Is that a fair statement?
Yeah, totally.
Yes.
Yep, totally.
And so, it's a whole different discipline than we're used to dealing with.
Oh, yeah, for sure.
And I don't know that I've read, this may be the only, or certainly the biggest systems biology paper I've ever read. This may be commonplace for that sort of data display, because they're dealing with, sifting through mountains of data that's both DNA sequence, RNA sequence, exome sequencing, mass spec data, and trying to make sense out of all of it. And this may be a fairly typical display and discussion. I don't know.
So the image that this conjures up in my mind, at least, is that we were always fond of saying, we can't fund this grant. It's just a fishing expedition. This is not a fishing expedition. This is a trawler that came up with all of the fish, and they're only looking at one species, and they throw all the rest out.
There's a whole lot of bycatch.
That is exactly what I was going to say.
I just wonder how the reviewers deal with this, right?
Right.
Because it would. I'm looking for the submitted and accepted dates, but I'm not finding them. I wonder how long it took to review this manuscript.
You certainly can't ask them to go back and do more experiments.
As a reviewer, I don't think a reviewer could go look at all of the, you're not going to look at the primary data. That's like doing the whole thing over again.
Exactly.
Right. So they received it December 12th and revised it March 5th. So the reviewers had it a couple of months, probably.
It's not excessive. Sometimes a year goes by, and but this is not excessive.
But this is meta-analysis, though, right? This is all based on everybody else's information, basically.
They did a lot of the experiments.
This initial laser capture, they did it with the Sinai Brain Bank. Later, they go to other collections to validate what they find. Yes.
And I think a key part of the reviewing is probably under the STAR methods. If you look at data and software availability, so they've put they say the viral abundance estimates and scripts to reproduce the results can be accessed here. So at least that part they have made publicly available. You can look at the viral part of the data and analyze it with the same scripts or with your own scripts. If you're in this field and you're doing that kind of work, you could probably analyze that.
Yep. They also found some microRNAs that are part of this AD gene networks, either lost or gained in preclinical AD. And, one of the miR-155 has been associated with innate immunity, malignancy, DNA virus replication. So they don't
even refer to the rest of the miR data.
No. I know.
So that really seems cherry picked.
They picked up one that they're very impressed with, miR-155. Yes, Dickson.
Yeah.
Yeah, what? Is it raining?
It isn't just raining. Okay. New Jersey just disappeared.
There you go. It's raining, Brianne.
That was a 15-minute delay from yours to us.
It took about a half hour because she said it was 15 minutes.
Okay, fine.
So that's the speed of travel from Madison to New York.
Another question about this paper is that they select single cells and do the RNA analysis on the single cells.
Yeah, laser capture.
Okay, now how many?
Is that cool or what?
It's cool, but how many single cells in a given area did they do to get an average?
Right. Read the methods. Dickson.
Exactly right. I mean, no, it wasn't. They didn't do it that way.
How do you know?
They didn't do it that way.
Well, this is. We've talked about this technique before. It was a paper from Charlie Rice's lab where they did this with liver, sections of liver in Hep C. So this is an okay thing.
Yeah, but when you're trying to make correlations between a generalized disease and a specific entity.
No, but I'm sure it's in the methods. I just have not focused on that.
You're very trusting.
I am very trusting. Absolutely. Otherwise, we'd never get a podcast a week, right?
And these are not and these are not random selected cells. These are specific cells. I want that one.
Here's the thing, Dickson.
How did you determine that one?
They looked at a specific region of the brain.
Yeah, go on.
And the controls, and then they selected cells within that region.
Yeah, so they have overall five different brain regions, and then, as Kathy said, 2 of them are mostly affected in Alzheimer's, but they look in multiple. And then later they look in different tissue banks from other people, and it kind of all agrees in the end, okay?
In the end.
There's some differences, and so that's why I think we don't have to go look at how many cells.
My sense, correct.
I just wanted to know.
Correct.
Go ahead.
My sense, correct me if I'm wrong, was one of the reasons that they did the laser capture and the single cell stuff was so that out of a brain section they could pick out neurons as opposed to other cells in the brain. Okay.
Mm-hmm.
It just raises a question about investigator bias.
Well, but you want to be biased in pulling out the diseased portions of the affected brain to compare them to the healthy portions of the control brain.
Well, I hope that was their response to the reviewers.
They're pretty honest about their approach here. Okay?
I don't doubt it. I'm not doubting it. I just think, when you look at what made you take that cell rather than this, or did you do a group of them and then average, or did you, that sort of thing. That's all.
So go ahead.
One thing that helped me out, Dickson, is that I had originally seen this paper on social media, and I saw another paper talked about around the same time. So I took a look at that one, and it turns out it was actually published with this one right behind it in the same issue of Neuron.
Mm-hmm.
And it does mouse experiments and cell culture experiments.
Cell cultures. Okay.
And so the two papers reference each other. So they clearly kind of knew what was going on, and there are quite a lot of things in agreement. I did find that one much easier to understand.
Okay.
Yeah, that's more of our kind of...
because you imagine yourself as the investigator. How would you proceed?
Yeah, well, this, as Rich said, this is a
difficult
approach that we don't do. And the other paper is more experimental, virology. Yep. So based on this initial pass of these gene networks, they said, okay, can we find viruses in these samples? So they do RNA-seq on these, again, the Alzheimer preclinical and the control brains from the Mount Sinai Brain Bank. And they do it from multiple brain regions. So we're looking at the RNA, the transcriptome, multiple brain regions. You sort through it and you say, what is there? And basically, in the end, 2 herpes viruses, human herpes virus 6A and human herpes virus 7, are the most consistently increased in terms of their transcriptome when you compare the disease versus the normal brain.
And in appropriate areas of the brain, right?
Yes,
right.
Appropriate areas for this disease, right. And if you could, I was looking for, the figures are presented in differential abundance. I was looking for raw numbers, but it's very hard to find. I'd probably have to go to the original data to find that.
You need even smaller type.
Even smaller type. And they even,
nano.
They get more granular. They go, what parts of each viral genome is transcribed in these tissues, and some are more than others. I don't think we have to get into that. But then they go to 3 other banks of brain samples that have been put together by different consortia. So get away from the Mount Sinai Brain Bank and look at others. One is the Religious Orders Study. One is the Memory and Aging Project, and another is the Mayo Temporal Cortex. It's 3 different groups, and they do the same thing, and they find essentially HHV 6A and 7 are up in these parts of the brain in the AD patients. And they do pick up some other viruses here and there, right? But these are the 2 that are consistently found in multiple places in multiple cohort collections.
And they're generally always picking up DNA viruses.
Right.
Yeah, with a few exceptions, which we'll get to later, right? They're mostly DNA viruses, right?
And they also looked, and I thought this was very important, they looked for HHV 6A and 7 in brains from the brain banks that had other forms of neurodegeneration, and they didn't find them.
Right.
So this is not just when the brain starts degenerating, these viruses reactivate. This is something that's unique about the Alzheimer's disease condition.
Exactly.
I just as an aside, I got interested in this religious orders study. I just want to read you this really quickly. The Religious Orders Study is a collaborative study with Rush and other U.S. medical centers. It involves more than 1,100 older religious clergy, nuns, priests, and brothers who have agreed to medical and psychological evaluation each year and brain donation after death. Researchers are using information from the study to discover what changes in the brain are responsible for memory and movement problems. Blah, blah, blah. I think that's terrific.
It's great.
Well, but we have to be careful with those data because, as we know, mice lie and monks exaggerate.
Oof. Oof. Oof.
Okay.
Excellent.
2, 3, 4.
I think it's great that so many people are willing to do this. Really,
Yeah.
I mean, you find that in a lot. The AIDS cohort, you find the similar thing, right? People are willing to do that. It's great. And they also find viral DNA. By the way, they look for that in these samples. Increased, in this case, 6. Okay. So now what does this mean?
Exactly.
We've got increased viral RNA. Of course, it could be, you got infected after the Alzheimer's. It made you more susceptible. It doesn't mean any causation, right? So they try and accumulate some data which would say there is an overlap of virus and Alzheimer's. And that's what the rest of the paper is really doing. So does the RNA abundance correlate with the traits observed in dementia and neuropathology, for example? And they can look at the levels of the transcripts and correlate them with the dementia score, the plaque density, and some other scores that are associated with Alzheimer's disease. And in fact, they do have a good correlation when they do that. And then they look at genes. They ask, we know that there are AD risk factor genes, right? There are quite a few of them. Do any of them overlap with the genes that here we see are regulating virus replication? And so here we have to bring up the concept of viral quantitative trait loci. These are regions of the genome that associate with viral abundance. That's transcript level. So they can say, okay, what parts of the genome are seem to correlate with higher viral abundance.
So if you have a particular, it's quantitative trait, because if you have a particular variant or allele at that location, you will tend to have more or less virus present.
So are you talking about host genes, QTLs, that are associated with viruses?
Yes.
Okay.
Right. Yes.
So it's a little bit confusing the way it's presented. Then
totally,
they're called virus QTLs.
And I will just warn anyone who goes to look at this paper. This is about the point where my student walked into my office, saw the paper on my computer, and said, "Why are you looking at a cyclone of viruses?"
Yes.
Yes, this is a cyclone figure there. Yes. And then you say, are there any AD risk factors in these regions that you identify as these quantitative trait loci? And the answer is yes, that there are AD risk factor genes in these areas that appear to be regulating viral transcripts. And they also say that maybe these genes are actually affecting nearby host genes, right? They're indirect effects. These are called cis QTL associations. Dickson, be careful. Don't knock your head on the table.
No, I'm not. I'm bending over backwards, in fact, trying to understand this.
So I think the bottom line here is that you have these AD gene susceptibility databases, and you have these regions that they've identified as playing a role in virus transcript abundance, and there's an overlap between the two.
Right. So the more virus you have in your brain, if you have Alzheimer's disease, the more virus you have in your brain, the worse the disease tends to be, or vice versa, because we can't be absolutely certain of the causality. At the same time, if you carry variants of genes that correlate with higher viral loads, you are more likely to develop worse disease.
Mm-hmm.
It's all tying together very nicely that especially HHV6A and HHV7 have this very strong linkage with the development and the severity of the Alzheimer's disease.
So the big questions are: does everybody carry herpes 6A and 7?
85% of the population.
85%.
I think that's the figure.
It's pretty high. Most people are infected.
It's extremely high. So that means that these host genes probably are more important than whether you had the virus or not. The subset of those people are going to be the ones that are going to develop Alzheimer's.
So here's the issue.
Yes.
Most of the population seems to be infected in childhood. In fact, these viruses cause
these are not congenital.
Rosebola. They're not congenital. They cause a rash disease of childhood, right?
Right.
But this is brain samples, right?
Yeah, I got it.
So I looked around. Apparently, quite a few people that get these infections later in life get CNS involvement. So these viruses apparently can get into the brain. Right? So, of the fraction of people that get these viruses, which is huge, what fraction have CNS infections, and what's the consequence there? They don't know the answer.
You should, if you have some genes that you can say are special to only these people, you can see where they are and then see if
that's the overlap. You have the overlap of these virus susceptibility genes and the AD susceptibility genes, right? And now the key is to figure out mechanistically what's going on. Try and develop models.
Why don't you just make a map as to where they overlap first?
Well, they did. That's what they have.
Throughout the world? They have a world
throughout
map?
the world.
No, I want a world map. I don't want this map. I want a world map of the 30 million people that have Alzheimer's?
Well, that's the the 30 million figure is going to be, is that U.S. or is that just developed countries?
No, that has to be the whole world.
50 million for the world now.
Okay, so say 50 million
all right,
people.
So 50 million known for the world.
So you take a random sample of those people and you match up their genomes with this study and you see whether or not the herpes virus overlaps there too. And then you've got a big, strong correlation. You got an epidemiological correlation.
I don't think we have a shortage of correlations here.
Yeah, no, I want something to hang my hat on here.
You're looking for a geographic association, but I'm
looking for a subset of people throughout the world. That's what I'm looking for, so that I can identify them before they even encounter herpes.
I think that's what they're doing with what they call viral quantitative trait loci.
That's per brain, though. That's per brain.
Yeah, but those are genes.
See what I'm saying?
Those are genes.
No, I understand that, but
so they're in the genome, so they're in all cells of the body, and these loci are alleles that correlate with higher viral loads during infection.
What about those?
So there's your population study.
And what about people who don't have herpes yet, who have these genes in that particular array? Do we know who they are?
Well, that's the problem. Part of the problem, because these infections are acquired in childhood, right?
Exactly.
So is that the point at which it goes in the CNS, and then many years later you have this result?
That's right.
Or does it stick around? Does it persist and recrudesce? We don't have any idea, right? Sure. I mean, these viruses establish latent lifelong infections, okay?
Right.
Like all the other herpes viruses. Yeah. And so the potential is that it's with you and you're fine. And then when you turn 50, 60, something happens, something in quotes, we don't know what, that then predisposes to Alzheimer's.
But you read a paper to me before our show started that indicated that the amyloid proteins are a defense response.
Well, that's the paper that Brianne was just talking about.
Prevents herpes from reproducing, but it's a plaque. After that,
so Brianne, why don't you summarize that paper?
Yes. So the other paper says that amyloid beta can be an antimicrobial peptide to protect against herpes viruses. And that herpes infection basically seeds or nucleates oligomerization of the amyloid beta. So the amyloid beta starts to come together in larger order clusters to basically trap the virus. Mm-hmm. And that will help block the virus. And so the idea is that there aren't these amyloid beta oligomers until virus infection begins that process. And they do some nice work with mice that overexpress amyloid beta and show that there are some changes in survival in response to herpes. They show that herpes infection leads to nucleation of these plaques, things like that.
So I got a really sort of simple-minded idea to sort of bring all this together. So this is my fairy tale, okay? So you get the herpes virus infection, you get virus in your brain, the cells are trying to respond with this amyloid plaque. It's a sort of an immune response, an innate immune response, if you like. Maybe, 99 times out of 100, that works fine. Okay, and you're protected. But if you have certain genetic characteristics, okay, that influence the plaque formation, maybe it gets out of control, and you wind up with abundant plaque formation and Alzheimer's disease. That would be the link between the Alzheimer's trait loci and the virus infection.
Exactly.
I was thinking of this as kind of like a tuberculosis of the brain situation, where you get the virus, it goes to the brain, like as Rich was describing, and it's there, and it's maybe inhibited by the amyloid protein and other factors. And when you're a kid and growing up, it's all fine. It's keeping it in check. The whole thing's sealed away. But then as you get older and your immunity starts to wane, it starts to go a little astray. And in some cases, you get these plaques building up, and the virus becomes more active, and reactivation of disease.
Right.
Yeah, I think something like that might be going on. If you look at the paper that we've been talking about with all of the QTLs, they reference the other paper somewhere, and they talk about viral entombment, which I think is a great term.
Mm-hmm. But the key here is that it's not just simply infection and defense with amyloid, there's got to be some other risk associated genes. And there may be more than one, because not everyone that gets HHV6, 7 gets Alzheimer's, right?
Right.
Exactly. That was my original point.
Also, this discussion that we're having is, an attempt. It's grossly oversimplifying and extrapolating on these papers. Okay, but at least it's a way to think about it.
Did you ever read the parable of the blind man and the elephant? This is this
is reductionist science. You have to simplify it.
You have to try.
Right. Otherwise, it's too complicated.
Also, these stories, these concepts that we're making up here with viral entombment and then subsequent reactivation, this presents a whole bunch of testable hypotheses that you can then go and look at. Well, if you do have viral entombment going on, you should be able to find the virus in there in some sort of interaction that's in some sort of tomb.
Some of that is in the other paper.
Is it right?
Although I will say the other paper largely looks at HSV1. They talk a bit about HHV6, but say that there's not a good mouse model of that.
Not a good mouse model.
So this paper unravels lots of potential interactions between virus and cellular genes, some of which are AD risk factors, some of which are antiviral. And I think that is all great future experimentation that people can do in cell culture or animal models. I wanted to highlight one of these where they did do an experiment. So they found this miR-155, a microRNA, a host microRNA that is a AD risk factor, and it's also, as I said before, has some antiviral properties in the literature. The HHV6A appears to suppress miR-155. So they have a mouse model. These are mice that have two human proteins, amyloid precursor protein and presenilin. They develop plaques, cortical amyloid plaques. It's a model for Alzheimer's, so they're transgenic mice. They cross them with mice lacking miR-155. All right. And they have more plaques and they're bigger. So miR-155 is somehow mitigating AD, and if you take it away, it gets worse. So the idea is that maybe the virus is downregulating miR-155, and that could contribute to,
So what's the test for this? The mice forget where the cheese is?
Plaques in the brain. They actually have some behavior.
Do they have characteristics that the mice exhibit?
They have some behavioral things, right?
Yeah. There are quite a few behavioral things that our neuroscientists here do. They usually do them with rats instead of mice, but there are quite a few learning and spatial memory tests you can do.
Yeah.
Okay.
So this
you can take them to the opera.
You could.
Yes, you could take them to the opera. So this is an example of what you could do for many of these genes identified. You could say, okay, if we had, and it would be great if they could infect the mice on top of it, right? I'm not sure that they can do that. So there's a lot more in this paper. They dig down into transcription factor regulatory networks. They identify metabolic areas that could be perturbed by virus infection that have been reported to be altered in Alzheimer's disease. For example, nucleotide pool metabolism and so forth. But the point is that looking at this in a big picture fashion, you can see that there is potential, as the title says, there's disruption of networks by this virus that are involved in AD. And I think that's the bottom line here. Now, I think it's very interesting that in the discussion, they say these findings do not demonstrate that virus contributes to the onset or progression of Alzheimer's disease. They say this could only be established in a prospective intervention-based study. So what would that be? A prospective study, of course, is one where you take two groups at risk for something and you give one a treatment and the other not. And of course, there are antivirals for these two herpes viruses.
No vaccines?
There are no vaccines, but we do have antivirals that are used. So the question is, what kind of study do you do? Do you take people... I don't know.
Teenagers.
I mean, no,
you take people who are 3 years old and start with acyclovir.
No, you can't do that. That's too much. You can't give people acyclovir their whole life. But could you take people with signs of AD and treat them?
Right. So I think the first experiment to do would be to try treating people who have AD. It's going to be an easier population to identify. But then from there, you'd probably go with the MCI population, the mild cognitive impairment population meant at the beginning. Try prophylactically treating a huge cohort of them. And obviously there are issues with that. You're going to be drugging a large number of people for something that you don't know will work, but see if you see statistical differences in a placebo-controlled trial with that kind of setup.
Yeah, I think it's very hard.
But that's going to take years.
It's
one of the biggest problems with Alzheimer's disease research is that it takes decades for this thing to develop, and it takes years once it starts to develop to really reach a point that you can say definitively that this is what's going on.
There's a wonderful movie that probably everybody's heard about and perhaps we've all seen. It's called Away from Her. Yeah. And it's a remarkable movie. It really is a great characterization of Alzheimer's disease during the early onset, middle part, and late stages. And it's well acted too. So I recommend it. That's not my pick of the week.
The problem with this interventional study is that it may be too late by the time you have symptoms, right?
Well, is it reversible? Is it reversible?
I don't know. That's a good question. It depends how much.
And is it part of that virus you had when you were a kid?
Exactly.
Yeah. It's all tough questions. So I would never start a study because I would have no idea. And of course, the clinicians would go, just try it.
Yeah.
Design a good study. And that's why these things get done.
We should have Rick Mayhew on our show because he's instrumental in those genes that are at risk for Alzheimer's.
Well, I would say, okay, there's some suggestion that these viruses may be involved. What kind of clinical study would you design?
Exactly.
Because here we are.
So the causality issue we can't nail down, but as they say in the paper, we report multiple streams of indirect evidence that
multiple
all
streams
point in the same direction.
It's a great phrase.
Yes.
And in the end, wouldn't it be amazing if acyclovir or ganciclovir reversed Alzheimer's?
Of course.
Or
wouldn't it be a miracle if we could find out that ulcers was caused by a bacteria?
So wait, one at a time. Let Alan first.
Yeah.
If a vaccine against HHV 6 and 7 could prevent it.
Yes.
Yeah.
Wouldn't it be a miracle if we could cure ulcers by giving an antibiotic?
Wow, that's amazing, Dickson.
I bet you that deserves a Nobel.
But Alan, your idea for the vaccine, you would first have to show that interventional treatment had an effect, right?
Yes. We're several steps before that at this point.
But then everyone would not take that vaccine, I'm sure.
Of course not. You'd still have idiots, but we don't have a vaccine against that, unfortunately, but maybe you could get most of the people protected.
Isn't it funny that the reason we have Alzheimer's is because of science, public health measures, vaccines, antimicrobials, right?
We're
living longer.
That's right.
Well, it's been with us probably for all of our existence, but we're only noticing it because enough people are living to those ages.
Exactly.
So I think today we've learned a lot about recrudescence and what happens when you have a lot of people living with something.
Yes.
Right. Yeah.
Did we ever mention the authors of this paper?
Yeah, I believe so. Did we?
Okay.
I think so.
There were numbers of people.
Let me look at them.
Ben Reedhead. And,
Reedhead,
Jean Vianney, Barry Mirand are co-first authors. Is that right?
Mm-hmm.
And then, so we did the authors.
Okay, let's read just a few emails here, just like 2 or 3.
And Norwalk virus comes from
Ohio.
Brianne, can you take Vincent's?
Sure. Vincent writes, Dear TWiV team, I wanted to write you all a quick note because Monday, July 30th is a special day. In TWiV episode 491, The Ilium and the Odyssey, you discussed a paper in Science pertaining to norovirus tropism for tuft cells. In this episode, you speculated on certain parts of the paper where there appeared to be missing information due to the page limitations. In episode 492, titled CRISPR Diagnostics, Dr. Craig Weiland wrote in to further clarify some of the details that had to be left out in order to fit the 6-page limit. In the end of his email, he mentioned that he will be moving to Yale to start his own lab and that he is looking for people interested in researching murine norovirus in tuft cells. After about a week of debating whether to email Dr. Weiland, I did. The same day I emailed Dr. Weiland, I had also just received my last rejection letter from the PhD programs I applied to previously in the fall. I remember vividly the day I received the email he sent back, rather quickly from my initial email. I was sitting in my apartment and wondering what the next step in my life would be and if this was a path I should continue pursuing. After a very pleasant conversation over the phone, he asked if he could contact my master's thesis committee at Quinnipiac University, pretty much up the street from Yale, and hired me shortly after as a postgraduate assistant. Monday, June 30th is my start date with him, and frankly, I am a tad nervous, but mostly excited. I am incredibly grateful for all the work you do to keep this podcast going and being a platform for great minds coming together all over the world to discuss the wonderful world of viruses. My closest friend Andrew listens to TWiV every week as well, and we discuss the papers together, as well as excitedly exchange other cool virology papers we find in our work. I promise I will work hard for Dr. Weiland so I can make him and the TWiV team proud. Thanks for being the best podcast in the world, Vincent Graziano. And Vincent says, P.S. If any of you find yourselves in New Haven, please do not hesitate to shoot me an email. New Haven has the best pizza in the country. Sorry, New Yorkers, it's true. And I'm buying you all a pie.
Okay,
I'll take the pie, but we'll have an argument.
Yeah, we are going to have an argument.
A discussion, please, a discussion.
But I think it was Kathy who commented, this is a great TWiV bump story.
Yeah.
And I agree.
Yeah. Great story.
Yeah.
I love his name.
Yeah.
And he just started on Monday of this week, July 30th.
Yes, yeah. July 30th.
Good luck, yeah.
And so New Haven, first of all, does not have pizza. That is not a dish that's available in New Haven. They have a pizza. And I don't know what that is, but I know that it's a circular thing that for some bizarre reason I cannot comprehend is cut into squares.
Really?
Pizza is cut radially.
Yeah, that's right. Yeah. Sicilian pizza is cut into squares, right?
That's right.
Yeah, but it's a rectangular pie.
Yeah, it's true. It is. Yeah, it is.
Indiana pizza is also cut into squares.
Yeah, and it's a rectangle to begin with.
It's got ham and pineapple.
No, it's round.
It's round.
Oh, it's round and they cut it into squares? Okay, then that's, yeah.
They do that on purpose to be different, right?
Yeah.
Wow.
So, and I had fun meeting Craig Weiland at ASV. I don't know if you did, Vincent.
Did not have a chance, no.
Yeah.
Cool.
Yep.
Well, that's a great story. Good luck there, Vincent Graziano. What a great name.
Vincent.
What a great name. Anthony sends us a link to a article. You know, when the U.S. was trying to promote polio immunization, they got Elvis involved, and they're digging him up again to try and get people, so to speak, to try and... All right, is there anyone who has not read an email? We have all, right?
I don't think I've read an email. Have I read an email?
All right.
I don't think so.
All right. Read.
Wait, before you do that, Anthony also has this refutations to anti-vaccine memes, which is a Facebook link with all kinds of things you can
people,
click on.
Yes, that's very good. Yes, I forgot to mention that. Exactly. It's very nice. And Rich, why don't you take Anonymous's email?
Okay. If I can find it here, because I just was panicking. Oh, there we go. Anonymous writes, Dear TWivers, a few points that I think are needed to be said concerning finding a postdoctoral position, and your comments and reflections will be highly appreciated. I'm defending my PhD thesis soon and looking for an appropriate postdoctoral position. My approach is a very simple one, sending emails. I do send emails to potential investigators where my scientific interests could be matched with a type of work they're doing. Actually, I do not know any other way of finding a position other than this. I recently read a blog post over Twitter about a PhD candidate that gave up on her PhD in neurosciences because of her supervisor's attitude towards her after over 5 years of investing work and effort in her research. I was not surprised. Supervisors that are not supervising and just staying in the way of their students is a reality that we all know exists in academia. It is just ignored and not really confronted. What concerns me here is that such PIs could not only ruin your scientific career while working with them, but even afterwards by writing unsatisfactory recommendation letters. New accepting PIs are understandably afraid of investing money and effort on someone that is not well recommended from his prior supervisor. It's like a judge hearing one side of the story and taking directly a decision. So you receive an apologizing reply or most times nothing. This needs to be changed. I do not know how. What I do believe is happening nowadays, more and more, and probably to circumvent such a problem, is that people are shuffling through labs because they already know each other or because they like each other, and through this they could minimize unknown aspects of the applicants they accept in their labs. Scientific competence is not a real player in such a model. I do like you. I get you.
I don't know.
Another important thing, and I'm currently confronted with and not fully understand, is why some researchers do not take time to reply to my emails. It is not that I'm sending a spam email or some impossible request. It's a very simple request. Do you have a place for me as a postdoc? Yes or no. It's not complicated at all, but it seems still for some human beings to be so tedious writing back, say yes or no. They forget, however, that it is very decisive for the applicant and for his family to receive such replies, that this is a hard time for the whole family, that it involves planning, it sometimes requires relocating to another country, and so on. I do not simply believe that these researchers are so busy replying to such emails, as some of them are simply tweeting every second, every minute, and sometimes only silly jokes. Feeling a responsibility here, saving families of poor young researchers, I would like to help established scientists answer such requests by copying one of these responses so that they do not waste their valuable time. Yes, I have a vacancy. No, I do not have a vacancy. Or 3, my partner left me and I cry all night, so I need some time to reply to your email, but I will when I'm over this, and directly after I finish writing some jokes over Twitter. At least then I know I would receive a reply in the future. I have a lot to say, obviously, but I would like to enjoy your discussion more than just reading my silent scream at you. Not so silent. So, geez.
Yep.
I have a couple of comments.
Mm-hmm.
Yep.
One is that when you have the unfortunate situation of having an advisor that maybe didn't supervise you all that well, that's what your committee is for. And so you have other people on your dissertation committee that are your advocates, or even other scientists in your department or your program that you should use as your advocates in that kind of situation. 2, depending on what people are putting in their cover letters, they might or might not get a lengthy and heartfelt response. But I do respond to everybody that writes to me. I have a simple script that I just copy and paste in if I don't have an opening. And related to that, in your cover letter, if you just tell about yourself and what you've done, that's not really going to get my attention. What's going to get my attention is if you say, Kathy, I'm interested in the projects in your lab. I'm especially interested in XYZ that you're doing, and here's what I think I could add to that. And so if I don't see that little kernel of interest in what I'm doing, I've had this feeling that the person is sending this out to 50 or 100 people, and they don't really care about me that much. And finally, you might be able to find something on the virology job site. So, little plug for the ASV virology job site. Just go to asv.org and you'll find a link over on the far right.
Well, I have a lot of people who don't answer my emails still after 57 years of being a scientist. So it starts at day 1 and it never stops because it's happened. And you have to be persistent. I find if you, after 3 or 4 or 5, if you don't get a response, maybe you have the wrong address. You better check that. But if you do repeat it, most of the time people say, I'm really sorry I overlooked it, because, it falls to the bottom of your list very quickly, your email, and they don't see it. So be persistent. I think Kathy's advice is great, but I would also say pick the right mentor to begin with. I think a committee is totally perfect, but try and get some information at the beginning. Talk to people in the lab, talk to other people to see, because, if you're picking a mentor in a program, you're there for a year before you have to pick your mentor. And so you can get a lot of information, and you will find people say, somebody came to me the other day and said, I did these 3 rotations, but I didn't like any of them. I didn't feel I would be a good fit, and that's good. So find some other place.
Well, that's not good, but that's a good thing to realize before you join one of those labs.
That's what I meant, yes.
Agreed. Yeah. I think everything Kathy said is spot on, both when I was applying for postdocs and from some of the emails that I receive. Doing your homework and showing that you've done your homework is important. As someone who does not have graduate students or postdocs, it can be really difficult to receive some of those emails when I know people haven't maybe done their research. And then, I've had one that said they wanted to be a great guy like me, which is a little confusing.
Great. Love it. Anything else?
Well, as old school as I am, I think I can recall the days when the person you got your PhD from would call a friend. Tell them what you want to do and where you want to go, and they call that person and say, I got a real good graduate student here, and I think he would be terrific in your lab. And the next thing you know, you've got the position. And that's actually what I did for my graduate students. I had one that went up to Harvard and worked with John David in the Department of Parasitology, and he worked on eosinophils and David Silberstein, and he had a great postdoc as a result of that. And it's because I knew that person, and I thought David was a good candidate and a good PhD student. And so just by word of mouth, a lot of this is done. So if you send somebody an email, it's the most impersonal way of receiving a request that I can possibly imagine. It's just out of the blue. And if it's out of the blue...
It's the way people do it now.
What if it's out of the blue? You're going to get out of the blue responses most of the time.
And
because
Dickson,
he's got a bad relationship with his mentor. That's the problem.
No, this person
that
isn't
is the
necessarily.
problem.
No,
wait a minute.
Person
What
is
if
describing somebody else who did. And if that your method will work if you can identify somebody who can be your advocate and call people in that same field. But if you're changing fields, then that can be more challenging. So I agree. That's the way that it's been successful for some people in the past. But people do write to people essentially like a cold call and say, here's why I'm interested in your lab and I want to do a postdoc.
The guy who told us his story, he got a research.
No. It was an unusual story, though, I think, and that's why we think it's unusual.
I changed fields and sent those emails.
Going to meetings and going to Gordon conferences and things of this sort when you're a student helps a huge amount. You get to meet these people.
I totally agree. I think face-to-face, we're not saying it's
the best.
It's not great, but nowadays the way to start a conversation is electronically. People don't telephone each other anymore. And out of convenience, it's okay because it's hard to get a cold call and you got to talk to someone about their career. It's better to get an email and return to it later. I think that works fine. But you do have to follow up with a face-to-face. I totally agree with you. And going to meetings, you can't beat that for sure.
And the email does have to be customized and indicate that you have some clue who you're addressing.
Yeah, I would say that if a test of this is that if you are sending the same email to multiple individuals, you're not doing the job right. Okay,
yeah, totally.
You're not even doing yourself a service. Okay?
You have no idea. Okay, go ahead.
You're not even that's an indication that you're not even really interested in the laboratory. You're just trying to get a job. Okay,
of course.
This is a matter of asking yourself what is it that I really want to do, and who can actually offer that to me. And when you figure that out, the customized email will be right there because you've done your homework.
Yeah, well, there was a time in my own life when I had my lab running that I got billions of emails from China. And, I've read all your papers, and I think you're a marvelous investigator. I'd love to come to work with you. I'm so and so and such and such. That's all they said. And then you send back, well, let me see some results from your own work. Or I would always email them back. But after a while, I just got tired of getting these things because I got like 50 a month or something like that. It was an incredible flow of information from people that I never even...
Is that why you're negative about emails?
I think that's the least popular way I have of communicating, I would say.
I still get emails. I just got one, I think, yesterday. My response, and I always respond, but my response now is real quick. It says, I'm retired.
That's right. I've closed my laboratory. I'm no longer taking graduate students. Thank you for your inquiry. I always say that.
I get a lot, too, and I say I'm not taking students or postdocs, and one person replied and said, thank you. It's the end of an era.
Probably right.
Well, email is here to stay.
I know.
And we have to learn to use it and to fill in the blanks. Right?
I know.
Fill in the blanks. Let's do some picks of the week.
Okay.
Brianne, what do you have for us?
Yeah. I have an article that was on Nature's blog in the career column. It was just published a couple of days ago, called Why It Is Not a Failure to Leave Academia. I think we might agree with that quite a bit, but it's sad to say that there are still a few others who don't. The reason why I really like the article is that there are some good tips for students about how to approach a PhD if they're not interested in an academic position, but also some good advice for PIs about how they can help encourage their students and support their students in a diverse set of careers.
Yeah, I found the first sentence hurtful in a way, not to me, but for the writer. As a PhD student in my final year, I find it demoralizing and frustrating to be constantly reminded of the bleak job prospects in academia. You know, there's this need to, so we're always talking about making people aware of the situation, right? Mm-hmm. But on the other hand, it can demoralize people, right?
Yes.
Mm-hmm. So what do you have to have a balance there, right?
Right. Well, I think that one of the things that I really liked that the article was talking about is that we shouldn't always think of a PhD just as a way to prepare for an academic career, but really as a way to think about, a scientific way of thought and a different way of training. It's not just about training for academics.
For sure.
So I think they say it's a traineeship in scientific thinking. And I think it's important to remember that.
Or as Alan Dove would say, it's an education.
It is.
Not a training program.
Yes.
Alan, what do you have for us?
I have something I ate last week.
Please, no, I don't want it.
And it's repeating, did you say?
No. It's a website referring to something that I ate last week that I was very impressed with. It's called the Impossible Burger. This is the first all vegetable-based, all plant-based burger that I've ever had that actually tasted like a hamburger. And I would say I would rank it as being certainly better than any other veggie burger I've ever had, and better than probably more than half of the beef burgers that I've had.
Really?
Oh. This thing is actually pretty. It's not like the best hamburger you've ever had, but it is a credible patty of non-meat that's actually quite tasty, and I was thoroughly impressed. And apparently the secret ingredient to this that makes it possible, it's not just that they got the protein there and shaped them properly so it looks a little like ground beef. They also added heme.
Right. I've heard that. Yes. Heme. That's what does it.
And Worcestershire sauce.
It makes a huge difference. It actually tastes like beef.
Blood.
Yes.
And I think what's important about this is, of course, beef is a huge load on the environment, and the amount of it that we eat is a big part of why we're changing the climate and degrading the environment worldwide. Now, I personally do still eat beef from time to time, but I'm aware of the problems associated with it, and something like this points the way toward a better solution.
So nutritionally, it's equivalent?
They've got a whole bunch of information on the website about that. I think it's actually a pretty good deal nutritionally. I don't know if it's, for example, as good a source of B12. That's always a problem with pure plant-based diets. But there are other ways to get that.
Dixon, want to go get one?
I'm not advocating that.
Yeah, let's get out of here. Come on, you're making me hungry.
Sorry, you're not advocating what?
I'm not advocating everybody go pure vegan, because I'm not going to. But this is certainly a way to eat less beef while still enjoying a good meal.
Cool.
So, Alan, did you get this at a restaurant, or did you buy this in a store?
I got it at a restaurant. So I went to a restaurant in Northampton, a very good little burger joint in Northampton called Local Burger. And I didn't even realize that they had this. I'd heard of the Impossible Burger and thought, gee, I ought to get one of those, see what it's like. And I saw that this restaurant in Northampton had them, so I went ahead and tried one.
So was it advertised in the menu as the Impossible Burger?
Yes.
Okay. So I'm looking, they actually have a thing on the site that you picked where you can find places near you.
Yes.
Mm-hmm.
So I'm going to have to try this out.
24 for Austin. Well, when you get down to it,
there's a whole slug of them.
Yeah.
Lots of places near here.
Cool. All right. I'm hungry.
Yeah.
Oh, I see Buda on the map, but doesn't look like there's any in Buda.
No, I think I got to go to Austin for that.
Rich, what do you have?
Oh, my. Okay, I got to get back to it here. Oh, yeah. I have a follow-up pick. We've done that before. That's legit. Okay, we did that with a Martian. I think the Martian got about 6 picks. Last week, I picked a thing about our friend Nassine's son-in-law being involved in sending a project to the space station about growing algae in space. And this is another link to basically the same thing, except this is a really nice 5-minute video that describes the whole thing, and it interviews Mark. The guy's name is Mark Settles. He's a plant biologist at University of Florida, and it interviews him, and it has all sorts of peripheral stuff with nice pictures about how they send this stuff up, the coordination of the whole supply mission, and how they deal with it in space, and a little bit about what the project is about. And it's just a nice video about sending a research project to space. I thought it was cool. Actually,
one
of the best parts about it is they send, as a separate experiment, they send up a bunch of mice, and they call them moustronauts.
Yeah.
And I've been wondering, I wonder how you package mice to shoot them into space. And it must be something for the mice, because that's a lot of G's you experience on.
Yeah.
Did you say G's or cheese?
You put them in a 50 ml conical.
There you go.
With the tip cut out so they can breathe.
The tip off so they can breathe.
Is that right? Is that what you do?
Then you put a big rocket engine on the base of the conical.
You like the fuse and everything?
That would cradle them nicely, right? I mean, that's the way we used to bleed mice, okay?
It's true.
Tail bleed, you'd put the mouse in the tube, it could breathe, you cut out the tip so they could breathe, then you could, and they wouldn't be able to run away. But it seems like a good way to cuddle. The coolest part of this video is when he squeezes the wet paper towel and the water just slides over his skin, right?
Yeah,
and Kate told us about that last summer. How that happened.
Yeah, I think that video was even a pick all by itself. Who was that?
Yeah, it's Chris Had.
The Canadian. Yeah.
Yeah, maybe.
Right.
Dixon, what do you have?
I have 2 things. They're related. One is a video that was made some time ago, back in 2015, I think, by a group at UCLA, indicating very strongly that there is absolutely no difference between art and science, because they both encourage creativity, they encourage productivity, all kinds of things. You watch the video, it's kind of nice. But then I found, as a result of just sort of surfing, that there's a brand new museum that opened in Singapore called Art Science Museum. All one word, but the S in science is capitalized. And it's a fantastic looking building. I'm going to go there, I think, in November, so I get a chance to visit this facility. And it cuts across all fields of art and science, and it sort of confuses you as to what you're looking at, which you should be, of course. So I just thought it was nice to see the 2 things together.
I think let's have a discussion of art and science next time. I want to talk a little bit about it,
I would like
but
that.
There's not enough time now. But I'm a little bit puzzled. Okay,
so there's an art exhibit downstairs now in this building.
We went,
and you posted some of my pictures on Facebook,
Instagram,
and somebody actually responded, and they said, "Wow, another scientist that does art." Meaning me. Thank you for calling me a scientist now because I'm retired. But then they mentioned Feynman, Richard Feynman, and he was an artist as well.
Yeah. But that's art. That's scientists doing art. You're saying in this article, science and art are indistinct.
Parts of the same process.
Let's talk about that a little bit.
Sure.
For our snippet next time.
You bet.
Kathy, what do you have?
First, a correction. It was Chris Hadfield, not Hadley. I always got his name wrong. But anyway, I picked something relating to where I was last week. I was in Santa Fe, and we saw the opera Dr. Atomic, and the next day we went to Los Alamos. Which kind of completed the circle of the science story. And in one of the museums there, they had playing Isao Hashimoto's 14-minute video that he made in 2003, which is a history of nuclear explosions from 1945 to 1998. And I picked this site, which leads to the Vimeo, because the site describes a little bit about what's going on in the video. And maybe that's true on YouTube ones as well. But it's really quite sobering. And I just learned a lot in preparing to see the opera and also at Los Alamos. And this was a nice reminder of some of that.
Did you go to
the video is really something.
Did you go to the Atomic Bomb Museum?
Well, I went to there's two museums in Los Alamos itself, and we only had time to go to one, but there were special exhibits in Santa Fe relating to, in conjunction with the Dr. Atomic opera. So I'm not sure which one you're referring to.
It's the museum in Los Alamos that has both replicas of the atomic bombs that they dropped on Nagasaki and Hiroshima.
Then no, that was the one that we didn't go to. That's the Science Museum.
That would have freaked you out. It freaked me out. Something ridiculous.
Well, they had models and a lot of photos. So, yeah.
This was life-size models.
Yeah, there's a life-size model in the
Fat Man and Little Boy. I believe they were called Fat Man and Little Boy. And I'm a child of the 60s. I can remember as a child, like 5 years old, I think, or 6 years old, they actually broadcast from Bikini Atoll, one of these explosions that you featured on this film, that they would let you listen to it. Crazy.
Yeah.
There was an exhibit at MoMA here.
Frightening.
Last summer, someone had found all the old films that the U.S. had taken of different tests, right? And somehow he got them and put them all together. And you went into this room and it would just be playing on a loop. No sound, just it was so, like this. I mean, what do we do?
Yeah, that's right.
I have a pick. I want you to listen to 2 episodes of the podcast Revisionist History. This is by Malcolm Gladwell. Season 3, episodes 8 and 9, because they talk about a time when Dingell's committee was investigating science in this country.
Oh, yes.
And he calls it a period of insanity, because if you listen, you realize he had nothing. He had nothing against any of the investigators. It was all garbage. And these 2 episodes go through it. And it turns out he was a science writer for the Washington Post at the time, and I didn't really know this. So he followed these very carefully. This first one sets it up, The Imaginary Crimes of Margit Hamosh. And the second, Strong Verbs, Short Sentences, is mostly about Bernadine Healy. She was chair of NIH at the time, and she testified before Dingell's committee. And she was brilliant. Her testimony basically put an end to it all. And you have to listen, but you start with 8 because it sets it up beautifully. He really does a great job with this whole thing, telling stories. So, Revisionist History.
That's cool. Interesting. My cousin in Santa Fe had listened to this podcast and was telling me all about the Bernadine Healy one. So
it's really good.
And confirms that.
8 sets it up really well. And it's about a time when we went crazy. There was an epidemic of insanity.
I remember.
When was this?
It was right after.
Sounds like we're in another
1991 to 2.
Yes, right, exactly.
And this right after I entered graduate school.
This included the Baltimore investigation and a few others that you might not have heard about. But Dingell was just going crazy, and he had nothing. There was an example, a grant application from someone at the Cleveland Clinic. It had a typo in it, which was a typo, and this is what they were focusing on. And he told Healy, "You signed off on this with a typo in it." I mean, so just listen; it's great. And they reenact this hearing was not recorded, but the transcript is available, and they reenact it. It was really good. Anyway, our listener pick is from, I'm sorry, Brianne, you want to say?
Oh, I was just going to say, I guess I know what I'm listening to while I'm traveling this weekend.
You should. Yeah. They're pretty short, and his voice is great, really nicely produced. Does a good job. Our listener pick is from Mark, who writes, "Hello, congratulations on episode 510 consecutive shows from the road. Do you have any shows planned to record in the San Francisco Bay Area? If yes, I'd like to watch if they are in an open forum. Next summer, ASM Microbe is in San Francisco. So if they allow us to do a TWiV at TWIM, we'll be there, Mark. So we'll keep you posted. I'm making a variant of a listener pick. It's a listener submission. I've noticed a shift in some of the language you and the other hosts use in your friendly banter. I've updated my TWiV bingo card with some of these new phrases. Images of the cards are attached. For sport listeners should take a shot of their favorite adult beverage when they hear one of the bingo phrases used. And remember, don't drive after drinking. So Mark is our friend from California. Used to be with Drobo. That should ring some bells.
Yep.
He's updated his bingo card. So
it blinks some lights for me.
Here's a challenge. Get a bingo. You have to write in which episode you heard each of these phrases from. So you can get a diagonal or a cross, right? That's bingo.
So you can do it with multiple episodes?
Yep.
Okay.
Just have to put in the episode where you heard each one. And if first 5 entrants, I will send you a copy of Principles of Virology.
Wow.
I'm going to buy them and send them to
Vincent, that's a big investment.
This offer expires August 31st, 2018. It's
Better
not
hurry.
Forever.
Better
I'm not
hurry.
going to keep it forever. There you go. Thank you, Mark, for that. That's TWiV 505, Apple Podcasts, microbe.tv/TWiV, and your app on your phone or tablet that you listen to podcasts with. Just search for TWiV and subscribe. Send your questions and comments to twiv@microbe.tv. And if you'd like to support us, we have 155 supporters over at Patreon, microbe.tv/contribute. We have over 13,000 steady listeners. You can do the math.
And you can support us on Patreon for just $1 a month, which is bingo square.
Exactly. Exactly.
Dickson Despommier is at thelivingriver.org and parasiteswithoutborders.com. Thank you, Dickson.
Great. This was terrific.
The skies are clear.
The skies are clearing.
Brianne Barker is at Drew University. She's BioProf Barker on Twitter. Thanks, Brianne.
Thanks for having me. It was really great to be back, and the skies are clear here, too.
Yes, we know.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
And welcome back to you as well.
Thanks.
Rich Condit is an emeritus professor at the University of Florida in Gainesville. He resides in Austin, Texas, but currently he's in Bend, Oregon. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at turbidplaque.com. He's also on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. I'm at virology.ws. Thanks to ASM for their support and Ronald Jenkins for the music. We've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.